Hello friends, in the previous video we discussed about genetic mapping. Today we will discuss some of the questions related to genetic mapping. Let us see the first question. Determine the linear order of the genes B, C, D, where it is given that distance between gene B and C is 14 map units, distance between gene B and D is 5 map units, and distance between C and D is 9 map units. If double crossover do not occur, here there is no double crossover, then map distance are additive. If we see the data, B and C are farthest, that is distance between B and C is the maximum 14 map units, and distance between C and D is 9 map units. That means this gene C is nearer to D than B. So if we show it graphically, distance between B and C is 14 map units, and this gene C is nearer to D compared to B. Hence, gene D is in between B and C. Distance between C and D is 9 map units, and distance between D and B is 5 map units. If we add this 9 map units and 5 map unit, then we get 14 map unit, which is the distance between B and C. From this, it is clear that gene D is present between gene B and C. So, the gene order is B, D, C, and we can represent it graphically B, D, C. Let us see this second question. Two genes are located on the same chromosome and are 14 map units apart. An homozygous dominant individual is crossed to an homozygous recessive individual to produce heterozygous offspring for both the genes. Heterozygous offspring are then crossed to homozygous recessive individuals. If this cross produces 1000 offspring, what are the number of offspring with each of the 4 genotypes? Capital A, small a, capital B, small b, capital A, small b, small b, small a, capital B, small b, and small a, small b. So first, let us see what the question is telling. Here the parental generation, homozygous dominant and homozygous recessive is crossed. We get the F1 individual, which is heterozygous for both the genes. Then it is crossed with the homozygous recessive individual, and this is known as test cross. Then we get the F2 offsprings, which are of 4 types, or these have the 4 different genotypes. Among them, capital A, capital B, small b, and small b, small b offsprings are of parental phenotype, and capital A, small b, and small a, capital B, small b, these are recombinant type. And it is already mentioned that genes that are located on the same chromosome are 14 map units apart. So, map distance is 14 map units, which is also equal to recombinant frequency or recombinant percent. Map distance or recombinant percent is calculated by this formula: number of recombinant offspring by total number of offspring into 100. 14 is equal to number of recombinant offspring by 1000, which is total number of offspring, into 100. So the number of recombinant offspring is 140. Number of parental offspring will be 1000 minus 140 equal to 860. So what we have seen that there are 2 types of the parental offspring, hence number of each of the parental offspring is 860 by 2, that is equal to 430. Similarly, there are 2 types of the recombinants. So, number of each of the recombinants is equal to 140 divided by 2 is equals to 70. So, the number of the offsprings having the parental phenotype is 430 and 430, and the number of the offspring having the recombinant type is 70. In this way, we can calculate the number of offspring with each of the 4 genotypes. Now let us see this question. The cross between homozygous dominant and homozygous recessive is made, and the F1 is then test crossed. The progeny genotypes written as the gametic contributions of the heterozygous parent are in the following proportion. Capital E capital F is equals to 2 by 6, capital E small f is equals to 1 by 6, small e capital F is equals to 1 by 6, and small e small f is equals to 2 by 6. Determine the distance between E and F. From the question, parents homozygous dominant and homozygous recessive is crossed, and the F1 offspring that we get is heterozygous for both the alleles. Then it is test crossed with the homozygous recessive individual. F1 produces 4 types of the gametes. Among them, if we see this capital E capital F and small e small f is parental type, and small e capital F and capital E small f is recombinant type. So now we have to determine the distance between gene E and F. That means we have to determine map distance. We know map distance is equal to total number of recombinant by total number of offspring. In this case, gametes into 100. Here the recombinant gametes are 1 by 6 plus 1 by 6. Total number of gametes are 2 by 6 plus 2 by 6, which is the contribution of the parental gamete, plus 1 by 6 plus 1 by 6, which is the contribution of the recombinant gamete. So what we get? 33.3%, or map distance is equal to 33.3 map units. In this way, the distance between gene E and F is 33.3 map units. Now let us see this question. A cross is made between homozygous wild type female Drosophila and triple mutant males. The F1 females are test crossed with triple mutant males. This is the triple mutant males. The F2 phenotypic ratio are as follows. So this is the phenotypic ratio of F2 offspring, and we get 8 different types of the F2 offsprings, or 8 different genotypes of the F2 offspring. Total number of F2 offspring is 1000. Now we have to determine the gene order and also determine the map distances between gene loci. First of all, we will see what is the parental genotype. The one which is most frequent is the parental genotype. Here ABC and A plus B plus C plus is most frequent, and hence this is the parental genotype, A plus B plus C plus and ABC. Now we will determine the double crossover. The one which is least frequent is the double crossover. Here A plus BC and AB plus C plus is the least frequent, and hence it is the double crossover genotype. If we compare this genotype with the parental one, so there is the exchange of A plus and A, and the genotype is A, B plus C plus, A plus B, C. What we have seen in genetic mapping, that in the double crossover only the middle loci or the middle gene exchanges its place. Here A and A plus exchanges its position. What does this mean? That gene A is in between B and C, or gene A is in the middle of gene B and C. You can understand double crossing over in my previous video, genetic mapping. The link is given in the description box. So the gene order is BAC. It means gene A is in between B and C. Now we will determine the distance between map distance between gene B and A. It is equal to single crossing over between gene B and A plus single crossing over between gene B and A plus double crossing over plus double crossover by total number of the offspring, which is 1000 here, into 100. So during the single crossover between B and A, the position of A plus C plus is exchanged with AC, and we get the genotype B plus AC and BA plus C plus. Here we will refer the table. A, B plus C and A plus B, C plus are the single crossover between B and A. So 114 plus 103 plus 12 plus 3 by 1000 into 100, which is equal to 24.2 map units. So the distance between B and A is 24.2 map units. Now we will determine the distance between A and C, which is equal to single crossover between A and C plus single crossover between A and C plus double crossover by 1000 into 100. When there is single crossover between A and C, then C plus and C exchanges its position and we get the genotype B plus A plus C and BAC plus. So here A plus B plus C and ABC plus, these are the single crossover between gene A and C. Thus 68 plus 55 plus 12 plus 13 by 1000 into 100 is equals to 14.8 map units. Hence map distance between B and A is 24.2 map units and between A and C is 14.8 map units. We can represent it graphically. This is gene B, A and C. Distance between B and A is 24.2 map units and between A and C is 14.8 map units. And distance between B and C is 39 map units. One of the important concept or topic in genetic mapping is interference and coincidence. So what is this? Crossing over does not occur uniformly along a chromosome. We determine the map distance and on the basis of which we tell about the crossing over. But actually what happens, that formation of one chiasma reduces the probability of another chiasma formation in immediate vicinity of the first. If one chiasma is formed, then up to certain distance, the chances or the probability of forming the another chiasma is less. This is because the chromatids are unable to bend back upon themselves within a certain distance. During the chiasma formation, chromatids bend and forms the X-shaped structure. Again, within certain distance, it is not able to bend back and form the another X. And so, when one chiasma is formed, then another chiasma formation does not take place in its immediate vicinity. This tendency of one crossing over to interfere with the another crossing over is called interference. As a result of which, there are fewer double crossover than the expected, or in other words, fewer double crossover is observed than would be expected according to the true map distance. Interference is measured by coefficient of coincidence. In short, we write CC, and this coefficient of coincidence is equal to observed double crossover by expected double crossover. So interference plus coefficient of coincidence is equal to 1. If the CC is equal to 0, that means the interference is complete and there is no crossing over. If this CC is in between 0 and 1, then there is partial interference and fewer crossing over may take place. If CC is 1, That means there is no interference and all the expected double crossover are observed, or in other words, there will be equal number of observed double crossover as expected double crossover, or observed double crossover will be equal to expected double crossover, and hence according to this coefficient of coincidence will be 1. Now let us discuss some of the questions related to interference and coincidence. In the first example, it is given that observed double crossover is 33 and expected double crossover is 39, and we have to determine interference. We know that coefficient of coincidence is equals to observed double crossover by expected double crossover. 33 by 39, that is equal to 0.846. Now we have to determine interference, which is equal to 1 minus coefficient of coincidence. 1 minus 0.846, that is equal to 0.154, or in percentage we can write 15.4%. Example 2. Expected double crossover is given as 2.3% and coefficient of coincidence is 70%, or we can write as 0.7. We have to determine observed double crossover. We know coefficient of coincidence is equal to observed double crossover by expected double crossover. 0.7 equal to observed double crossover by 2.3. Therefore, observed double crossover is equal to 0.7 into 2.3, that is equal to 1.61%. This is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.